So we have discussed all the morphological changes that are taking place in the nucleus, in cytoplasm, in plasma membrane, in mitochondria, centrioles. So now we will discuss the head part of the mature spermatozoa. We will discuss the other three parts also of mature spermatozoa, that are the neck part, middle piece and the tail part. First we will discuss with the head part. So this is the head structure of the mature spermatozoa. It has the acrosome and the nucleus. So let's see. This central part, which have the two envelopes around it, this is the nuclear envelope, and the central part is known as the nucleus, this dotted structure. Around the nucleus we know cap-like structure, that is the acrosome. This is the acrosome part present around the nucleus, and around acrosome we are knowing that there is the plasma membrane. Okay, so this acrosome is also having the two membranes. First is the inner acrosomal membrane, second is the outer acrosomal membrane. And in acrosome we are knowing this dotted structures are the secretions of the proacrosomal vesicles, and here it is having this bar kind of structure. So let's see what is acrosome part and what is post-acrosomal region. You can see here acrosome is present till here, acrosome is not present after this, but nucleus is present after this. The part where nucleus is present but acrosome is not present, that part is known as the post-acrosomal region. And the part where acrosome is present, that part is known as the acrosome. And acrosome has the three segments. First is principal segment, equatorial segment and apical segment. This apical segment is the part of principal segment only. You can see this secretions one part is present till here, and after that bars kind of structure is present. The part that is having bar kind of structures, that part of the acrosome is known as equatorial region, and this secretions part is known as the principal segment. And you can see this tip, this rounded tip is present at the just tip of the nucleus, just above the nucleus. It is not present at the sides, and this part, this tip part is known as the apical segment, but this is the part of the principal segment. So equatorial segment is having these bar kinds of structure. So what is function of this bar kind of structure? These bars joins the outer acrosomal membrane and inner acrosomal membranes. It makes the link between these two acrosomal membrane. So this is the head part of the. So we have discussed that at the end of the or the posterior end of the nucleus there is present a centriole, which is helping in the formation of the flagellum, which will arise from the centriole and then move to the middle piece part and then to the tail part. So this is the TS of the flagellum. So the flagellum is having 9+2 arrangement. 9 is of the doublet and 2 is of the singlet. So let's see. These two circular parts which are present in the centers, these are the singlet microtubules, and these two singlet microtubules are connected with each other by the bridge. And around the singlet microtubule there are present these doublet microtubules, because two microtubules are connected with each other. So this part is doublet, 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 and these are the 9 doublet microtubules, and here these are the single microtubules. So let's see this. Can you differ in the structure of these two microtubules? In this microtubules there are some fibers present around this microtubule, and this microtubule do not have any kind of the fibers. These are the fibers which are present around this tubule, and this tubule do not have this kind of the fibers. And this is the sheath which is present around these two microtubules. So now these doublet microtubules are connected with this sheath by this link. This is known as the radial spoke. Radial spoke makes the link between this sheath and the doublet microtubules. And these doublet microtubules are covered by the membrane, and these are connected to this membrane by the membrane link. So let's come to the structure of doublet. In this doublet there are the two microtubules which differ from each other, because these microtubules do not have any kind of arms, but this microtubule have the two arms, one this arm and one this arm. This is the outer arm and this is the inner arm. You can see this is the outer arm and this is the inner arm. This is the inner arm and this is the outer arm. And these arms are absent in this microtubule. So what these arms are made of? These arms are made up of a protein that is the dynein protein. These arms are made up of protein that is dynein, D-Y-N-E-I-N protein. And what function is of this dynein protein? One more thing, these two microtubules differ from each other, so we give them name. The arm one microtubule is known as the tubule A and the without arm microtubule is known as the tubule B. So these arms in the microtubule A have the protein dynein, which act as the ATPase enzyme. So what is function of ATPase enzyme? That is, it converts the ATP into the ATP and release the energy. And this energy help in sliding of two microtubules over each other. In some cases, sperm is immotile, it is not able to swim, it is not able to move. So what is the reason behind it? In that case, the flagellum is not having the protein, that is the dynein protein. If there will be no protein, there will be no sliding of the microtubules. And how does there is no protein? Protein is formed from the mRNA and mRNA is formed from the DNA and DNA is having the genes. If any kind of mutation is taking place in that gene, there will be no transcription, there will be no mRNA, and if there will be no mRNA, there will be no translation and no protein will be formed. And if there will be no protein, then there will be no sliding of the microtubules. So in this structure, we will discuss three proteins. One we have discussed, that is the dynein protein, which is present in these arms of the subtubule A. Second is nexin protein, N-E-X-I-N. This protein is present between this interlink between these two doublets. These two doublets are interconnected by the link, that is inter-doublet link, and this link is made up of this nexin protein. And the very third protein which we are going to discuss is the tubulin. This tubulin protein helps in the formation of these microtubules. So we have discussed the structure of the flagellum, in which we have discussed this 9+2 arrangement, in which these are the two singlets present in the center and the 9 doublets which are present around the singlets. And these proteins we have discussed: nexin, tubulin and dynein protein. So we have discussed the head part. So this is the head part, assume it, and this is the next connecting piece from where the flagellum is starting, and it passes through the middle piece and then it reaches to the tail region. In the center of the middle piece and the tail region, there is present an annulus, a ring kind of the structure, and the tail region is having the two pieces. One is the principal piece and another is the end piece. Now flagellum is passing through middle piece and the tail piece, so we will discuss the structure of the flagellum in all these pieces. In the middle piece, we are knowing it speciality of the middle piece, that it is having the mitochondria around it. So let's see, in the center it is the singlet present, and then sheath is present around it, then the doublets are present. Around the doublets there are the 9 outer dense fibers, and around the outer dense fiber there is a mitochondrial sheath and the plasma membrane is present. The speciality is here that mitochondrial sheath is present, and another thing, that is the outer dense fibers are present. So this is the flagellum of the middle piece part. Then next is the principal piece part of the tail region. So let's see here. First of all, what do you see the difference? The size of this is less than the size of the flagellum of the middle piece. Second thing, you notice here that there is no mitochondria. Here is only the singlet present in the center, then the sheath is present, then doublets are present. One more thing, here there was the outer dense fibers, only the one layer of the dense fiber. Here are the two layers of the dense fiber, outer dense fiber and inner dense fiber, but there is no mitochondria and size is reduced than this. So next is the end piece part. You can see here it is the just normal flagellum like kind of the structure. Here is no mitochondria, no fibrous sheath, and size is very much reduced. Only the plasma membrane is present. So this is the structure of the mature spermatozoa head part, neck part or the connecting piece part where the centrioles are present, from where the flagellum is arising, and then middle piece part. Here is the annulus, the ring kind of structure present between these two parts, and then is the tail region. Here is the principal piece and here is the end piece. In middle piece there is the mitochondria, in no other piece there is the mitochondria, and centrioles are present in the part of the connecting piece or the neck part. So we have completed with the spermatogenesis here, and the mature sperm structure we have discussed.